Hello and welcome to the second of a series of 4 webinars about CRISPR-Cas, a very special tool for biologists that has been discovered some 8-9 years ago and has in this short time revolutionized the way life scientists work. These 4 webinars are organized by the Forum for Genetic Research from the Swiss Academy of Sciences, and they are co-sponsored by the Federal Office for the Environment. I am Thomas Häusler, a science journalist with Swiss Public Radio, and I'm moderating this event. In today's seminar, we will look at CRISPR and food production. That means we are going to hear about how scientists are using CRISPR-Cas to change genomes of plants and improve their properties. For instance, make them more resistant to pests or diseases, lower their need for water, or make them more tolerant to salt or drought. We are also looking at the implications this has for food production and how the public reacts to these developments. There will be 3 presentations in total and afterwards a Q&A session. As this webinar is not really a good format for a full-blown debate, we ask you to stick to questions rather than stating opinions on the subject. Also, please concentrate your questions to today's topic, CRISPR-Cas and food, as there will be additional webinars later about CRISPR in medicine and another one about societal aspects. Feel free to write your questions already during the presentations in the Q&A box. If there are questions of understanding, we will take those up directly after each talk. All the others will be answered in the Q&A session after the 3rd talk. Now we come to our first speaker, which is Dirk. He is the director of the Center for Plant Systems Biology of the Flemish Institute of Biotechnology. Dirk analyzes the networks that regulate plant organ growth and crop productivity. He will talk about how CRISPR-Cas is used by plant scientists to improve crops. Please, Dirk.
Well, thank you very much, Thomas, for this very kind introduction. Also, thanks to the Swiss Academy of Science for giving me the opportunity to present the impact of CRISPR-Cas on breeding and on food productivity. I will share my screen and voila, so that should and open it the presentation full. Thomas, do you see this? Hello? So I suppose that everybody sees all this now, and so the CRISPR-Cas is the holy grail of plant breeders, and you will see immediately why I make this statement. Well, first of all, I would like that you take a good look to the next picture. A picture that you all very much used to, that you find in most Western supermarkets. You see there plenty of food. It looks all very healthy. It's available at all times. You see a large amount of varieties there, and we take this for granted, but one has to realize that what you see there is actually the product of many years of work of plant breeders and plant breeding. And one cannot not really understand or to underappreciate the role of that in our food productivity systems. Most food are derived from wild plants here, and this is a classical example where you see on the left side of this picture, you see cabbages, Brussels sprout, kale, broccoli, cauliflower. You would think they're all different plants, in fact, but they do not occur in nature as such. They are all derived from Brassica oleracea. It's a wild species. You can still find in Europe, in some places. But through selection, through breeding, we have created this very large variety of different plants here. This breeding process takes many years. It takes selection, it takes trials like in what they call field trials like here. You see, for example, for one of the cereals like here. And this happens in many places all over the world. Like in fact, many companies who are doing this work, like in fact, have summer nurseries where they grow plants in summer, and they grow them also in the winter, our winter time, for example, in South America to further improve the breeding process. Now, for breeding is one important thing that you need. You need genetic variation. You need variation in the genes, and this variation can come into and can be achieved in many different ways. Like spontaneous mutations happen all the time, also in our body. Like in fact, but if you, for example, go to a wheat field in the summer. And you would look to the genetic information of every wheat plant, you will see that there are in this wheat plant, there in the genetic information, there will be mutations accumulated. They occur as part of our life, at a relatively low efficiency. You can further increase the genetic variation by doing crosses with wild relatives. For example, you can look for a relative, a poorly producing relative which has a certain disease resistance gene, and when the species can be crossed, you can cross in that genetic information and obtain new varieties, highly productive with resistant genes. Since the 40s of last century, also mutation breeding is very common. And the mutation breeding is in fact that you increase the genetic variation by in fact treating plants with DNA damaging chemicals or by irradiation. So this is being used, and currently in our food production, there are more than 3,000 varieties, and which are in fact produced in this way. For example, when you drink a beer, you're using barley varieties, which are obtained by mutation breed. It's a very common practice to increase the genetic variation. Much more recent technology, the typical GMO technology, where you introduce most of the time foreign DNA in a species, is another way to increase genetic variability. And this already relies on knowledge. You take a gene that you introduce. And as you all know, in Europe, this type of plant improvement is very strictly regulated. And now we have a new kit in town, which is called CRISPR-Cas based, and I call it precision breeding, because what essentially it's a tool to in fact to create mutations very much the same as you can spontaneously obtain, but now with a biotechnological method. The end product cannot be distinguished between spontaneous mutations and what you can obtain by CRISPR-Cas based methods. So I took this picture from, I think, a Swiss, I'm not sure, a Swiss newspaper here, which explains that essentially by crossing species, in fact, you can introduce new genetic variation or by mutation breeding. In both cases, in fact, this is not being regulated. And in the bottom of the picture, you see genetic variation which is caused by gene technology, either by introducing foreign DNA in red here, and that's a GMO, or by this CRISPR-Cas method, where essentially you make mutations exactly the same way that you would do by conventional breeding. Nevertheless, in fact, the European Court of Justice ruled that this mutation in the bottom of this picture obtained by CRISPR-Cas should be treated as GMO, although it's not distinguishable what you can obtain by classical methods. And that's the whole debate. I think that here with this scissors here, you can very targeted change the desired genetic information very fast, where in the other methods, it takes really, it's by luck, by chance, and it takes an enormous amount of time. So I summarized this in 2 slides. what I call is conventional breeding, which is not regulated. It's very time consuming. You look, we look for time periods, 5 to 25 years. I give you an example here later where it is even much more. It's non-targeted, so you really look it by chance, like in fact, and you can increase the probability, for example, by using mutagens. It's non-targeted, but you're really looking like for a needle in a haystack, the one in a million events, also, or even more, that give you the desired trait. Crops have often very complicated genomes, like wheat that you use for bread. It contains like 3 copies, actually 6 copies, because it's a diploid. For every just the hexaploid, so you need 6 genes for every trait. So you have to find alteration in all 6 genes before you can have a desired trait there, which is almost impossible to reach. Very often you also see that positive genes which contribute, for example, to disease resistance are linked to negative genes, genes which, for example, negatively affect yield. And the example I'm going to give you in the next slide is one of them. And they also require, because the long time you need it and the uncertainty of the events, it really requires a large investments. So one example of this time scale, for example, is late blight resistance in potatoes, a terrible disease which affects, kills not only the upper parts of the plants, but also makes that these potatoes are completely not eatable. It's a fungal disease. Well, finally in 2005, after 45 years of breeding, in fact, we're able to come up with a commercial variety of potato which have some resistance to this late blight. It's even not in the best performing varieties, because one of the problems is in this case is that negative genes are linked to genes which give resistance to this late blight. This process can be enormously speed up by genome editing. By CRISPR-based genome editing is very fast method. It's targeted. You go exactly. You make changes in the genes that you want to change. And it also, what is important to realize, it relies on a huge amount of information on the function of genes. Since the mid-70s, from the last century, enormous amount of work has been done by molecular biologists all over the world to decipher actually the function of genes. And this knowledge actually goes now into breeding. It's a relatively easy and cheap method, also accessible. And we see this by examples now in Argentina, for example, where this technology is already commercially applied, that is accessible to small companies, to small crops. Not only the big companies can do that. And it, in fact, it's what is important to realize, that in the end product, the finally improved crop is indistinguishable from crops developed by conventional method. If you don't know in what has been done, it's impossible, whatever method you're using, to figure out whether this is obtained by classical breeding, by conventional breeding, or by genome editing. And it's also for one of the major reasons why only 8 years after the discovery of the development of this method, that already the Nobel Prize was given to Emmanuel Charpentier and Jennifer Doudna, because this is absolutely revolutionizing the way that we can improve crops. Just 2 examples. There are many now in the literature and being commercially developed. Already said that wheat is a very difficult plant. In fact, and for example, there are genes now which makes wheat susceptible to this terrible fungal disease, powdery mildew. Essentially, when the plants are infected by that, it kills the wheat plant and reduces enormously the productivity. Well, this susceptibility makes wheat susceptible. This can be inactivated, but you need to inactivate all 6 genes in order to have the desired phenotype. This is almost not possible to do by classical breeding. It would require enormous investment. Nevertheless, with genome editing, this is relatively straightforward. These plants do not need fungicides anymore to really perform in the field conditions. Another example, again, using wheat, like to stay with this plant, like is the presence like, of certain proteins, so-called gliadins, like in wheat, which causes severe forms of allergy, which is known as celiac. Like in fact, there are many of these genes, like in fact, like in wheat, that makes like that for people who are susceptible for that. That makes that eating gluten-containing like wheat, this is gluten, like in fact, makes them like in fact like so susceptible to that. There are 45 genes, like in fact, like in wheat, like this, and by this would be impossible to change by classical breeding. Well, but genome editing groups already a couple of years ago, in fact, succeeded like by to have 35 of the 45 genes modified, which caused like a very strong reduction in immunoreactivity, and thereby, like in fact, much better for people actually to eat. Certainly, people who are sensitive to gluten. So what can genome editing deliver? I showed already an example of the pathogen and pest-resistant varieties, so that would lead to less pesticide and a more eco-friendly agriculture. But it also can improve nutritional qualities, more unsaturated fatty acid, low gluten wheat. Japan is now bringing a CRISPR-based tomato on the market, which is reducing blood pressure in elderly people. You have improved nutrient use efficiency. So, for example, by modifying the root systems, to have deeper roots, for example, which leads to less eutrophication and lower release of nitrous oxide, which is a very strong greenhouse gas. Resilience to drought and to heat, which in view of climate change will become more and more important. But also higher yields. In fact, the idea to improve in a sustainable and eco-friendly manner yield is a good thing, because it actually allows us to use the land that you not use, like for example, for renaturing and for preserving nature. We can also think about the improvement of protein crops for Europe. Now Europe is importing huge amount of mainly soybean as feed, mainly GMO products, in fact. But with genome editing, we can much faster breed for protein crops, and maybe thereby reducing the impact on climate as well. But also we can improve more varieties, improvement of old varieties. And so that's, and for example, if you think about enormous problem with grapes, grapes you cannot breed because you lose all the favors. If you have Sangiovese as a variety, you cannot breed for it because that the variety will be lost. But with genome editing, we can make fungal resistant Sangiovese grapes, and keeping all the flavors and the qualities of these grapes, as an example. We can also develop new crops, crops, in fact, which are now maybe somewhat not used because they're not very productive, but there's enormous variety out there in nature. And we know many genes which are involved in this, what we call domestication. Well, maybe by genome editing, we can improve this domestication process, and having these new crops also introduced in our agriculture system, and thereby improving the resilience of our agriculture systems. So many science organizations, in fact, in Europe, have made statements concerning genome editing. These are science-based statements, in fact, so which all say that this is a fantastic method that one should really further look into and to deregulate, to some extent, in Europe. So this includes the group of chief scientific advisors of the EU. It includes large funding organization, the Max Planck Institute, the Leopoldina, the European academies, but also the farmer organization, European farmers and the European agri-cooperatives, the COPA-COGECA group here, companies which are assembled in the Euroseeds breeding companies. It's Epsom and also Alea. That's another organization with more than 50 academies involved. We have the European, the ethics group of ethics and science and technology, plans for the future, and so on. And also, I'll show you that in a minute. EU-SAGE, stands for European Sustainable Agriculture through Genome Editing. But I'll come back to that in the next slide. Furthermore, we see on a world scale, in fact, that only Europe and New Zealand have for a moment and legislation which makes actually the use of this technology for agriculture improvement not possible. So it's really even countries which were traditional very much against GMO, for example, like Japan, even have already legislation for the use of this technology in place. And this is particularly important for breeding. As already said in the beginning, breeders are doing that in Europe. They're using the summer period to look for improved crops, and then they move in our winter. They move, for example, to Chile or to Argentina to further improve. So this is a global way of improving crops. The fact that we regulate now genome editing crops like in Europe means that all these improvements like here will not enter the European market anymore, will not enter the European breeding efforts anymore, and we will actually not be able to use this tremendous opportunities. So this is also the opinion, and this is by consensus, of all European scientists, and in fact, which are working on this molecular aspects of plant improvement. So they are now brought together in EU-SAGE, and these are scientists, in fact, they are altogether now 134 working at 134 different science, plant science organization across Europe. So all of these scientists, they agree with the fact that genome editing, which does not add foreign DNA to a crop, should be exempted from this GMO legislation. And then let me close with a final slide, just saying, and my slides do not move anymore, but now it works. So my final slide is, if the future could whisper, it would call out for CRISPR. And with this, I would like to thank you for your attention. Thank you.
Thank you, Dirk, for an excellent talk. Thanks so much. A lot of food for thought, I think. And there is already a first question in the Q&A box. Thanks for that. We will, since it's a general question, answer that in the Q&A session later on. If there are questions of understanding, please enter them now so we can, ask Dirk directly. And I wait for a few seconds if that happens. If not, we will go on. Looks like you're okay. No, that's also one for the Q&A session. So it looks like you had just were giving a very clear talk. No questions of understanding. Thanks so much, Dirk, again. So we come to our second speaker, and that is Matin Qaim, and he is Professor of Agricultural Economics at the University of Goettingen. And his research is about sustainable agriculture, food security, and rural development. Matin has worked in numerous countries in Africa, Asia, Europe, and the Americas, and he will talk about important challenges for food and agriculture, like food security, and staying within the boundaries of the planet, which, as we all know, are kind of scratched already. And also he looks at what role gene editing can play to help address these. So please, Matin, go ahead with your talk.
Yeah, thanks, Thomas. I'm happy to contribute to this seminar here. I should mention that I'm not a plant breeder, I'm not a biotechnologist, but I look at this from a social science and economics perspective. So let me start by talking some of the global challenges for food and agriculture. Where do we stand? And it's important to not only look at Europe, but to understand that we are actually on one planet and the problems of the planet are relevant for all. And from a European perspective, we often do not primarily think of having too little, but of the world population, around 1/4 does not have enough calories or nutrients. And this is obviously particularly happening in developing countries of Africa, Asia, and Latin America. If we take a closer look, we also see that, well, this is not only about having too little food production. It's related to poverty. It's related to the fact that many people are just too poor to afford enough food and food quantity and quality. But we also have to realize that these people often do not only depend on agriculture and food production as a source of food, but as a source of livelihood. Many of these poor and undernourished people are actually farmers, farming themselves or working in farming. So there's a very close connection between small-scale farming and undernutrition. If we look into the future, of course, we all know that the population continues to grow. And the population growth is not happening in the rich world, it's happening in the poor countries that are undernourished anyway. We are likely to see 10 billion by mid-century, and this is happening against planetary boundaries, limited water, limited land, and limited other resources. We are already having a situation where agriculture contributes significantly to environmental degradation, to climate degradation. And this global environmental change is not only a problem per se, but it's having feedback loops to agriculture. We know that agriculture is affecting, climate change is affecting agriculture production negatively. We have experienced that in Europe over the last few years, but this world map here shows where the effects of climate change on agriculture will be most dramatic. And this is where you see the red color in the map. And this is mostly in the global south, where many poor people depend on agriculture. So this is going to have feedback loops and will likely increase poverty and undernutrition if you're not acting and acting rapidly. So there are a number of things that need to be done. We need major food system transformations in order to make agriculture and nutrition more sustainable. And it cannot be done with a single technology. It cannot be done with isolated steps. Many things are required, and many of them are unrelated to technology. But if you're looking closer, then we see, well, there are a couple of things, quite a few actually, where technology can help, and plant breeding in particular could help in terms of increasing production, reducing the environmental footprint, reducing poverty, and so on and so forth. So breeding matters. And this slide is showing a bunch of different breeding methods. Dirk has talked about some of them in much more detail than what I want to do. I just want to say, isn't that great that we are having an additional number of tools in the toolbox of breeders, and that things are becoming ever more precise and ever more efficient? I think every plumber would be happy if they would have increasingly precise tools in their toolbox, and this is certainly true also for plant breeders. The only thing, and Dirk also mentioned this, is that there is an artificial divide here where you see the dotted line. Everything on top of the dotted line is considered conventional breeding, or is termed conventional breeding in the public framing, and everything below is considered as genetic engineering, resulting in genetically modified organisms. This is not only a wording, but it's deeply rooted in beliefs, because conventional breeding is commonly considered by the wider public as something that's natural and safe. and genetic engineering is often perceived as unnatural and risky. And this is also Europe's regulatory approach. I mean, the techniques here are regulated in a very different way, much more strictly than what we have up here. And this in spite of the fact that we know from over 30 years of research that GMO crops are as safe as conventional ones. This is holding true for transgenic GMOs and even more so for gene-edited crops that do not contain foreign DNA. So we are seeing regulatory approaches and beliefs that are not based on the scientific evidence that we have. But the public debate is broader than just immediate environmental and health risks. There are broad concerns, social concerns related to high-tech farming. And this is related to some romantic view of how agriculture should look like. So people are very concerned about increasing use of chemicals, about less diversity and increasing monocultures, increasing farm sizes, huge machineries that are being used. People are concerned about patents on life and market monopolies of some multinationals. In short, people are concerned about industrializing food systems. And indeed, we see such tendencies, and indeed, many of them are problematic. The only question we need to ask ourselves is it not too simplistic to assume that all new technologies will necessarily further aggravate these issues? And the answer is no, they won't. I mean, Dirk has mentioned a lot of what is already being done. I mean, gene editing can actually develop resistances against pests and diseases such that we can use lower chemical pesticides. Gene editing can help to produce nitrogen and phosphorus efficient plants such that you can use less chemical fertilizers. There is successful work in already more than 40 different crop species. So it's not only soybean, it's not only maize, but really a large number of species that breeders already work on. In terms of farm size, well, seed is a very traditional input that every farmer uses regardless of big or small. And obviously, gene editing, this is also what we know, is a relatively cheap technology that can not only be used by large companies, that can also be used by small laboratories, by small and medium-sized enterprises, if efficiently regulated and managed. And this is, of course, a big if. So we should much more talk about these things of how to regulate efficiently rather than to have the black and white question, is the technology as such good or bad? In this slide, I want to further emphasize the statement that it's not the breeding methods that matters, but it's the crop traits and how they're used in farming and by countries around the world that in the end determine the effects of the technology. Most of you will know that the world has been using transgenic genetically modified organisms already for more than 25 years, not much in Europe, but in other parts of the world. And there's already quite a bit of evidence about the effects. This is data from a meta-analysis that we carried out with data from 25 different countries on the two main traits, insect resistance and herbicide tolerance. And we see that both types of traits lead to yield increases, farmers benefit in terms of higher profits, but we see quite a significant difference in terms of how they influence chemical pesticide use. Insect resistance reduces, whereas herbicide tolerance does not reduce chemical pesticide use. So it's not dependent, both are GMOs, but it's not depending on the breeding method. It's depending on, we're talking about two different traits. There are also differences in terms of this technology that's successfully used by smallholders, whereas this technology, which is so far at least mostly used by large-scale farmers in terms of soybean monocultures. So in the end, we need very serious debates about what type of agriculture, what type of traits we want and need for sustainable farming. But this is much more about these types of debates rather than trying to block useful technologies, which is really counterproductive. Let me say a few more words on smallholders and new breeding technologies. There are a number of developing countries where insect-resistant, so-called BT crops have already been used for a larger number of years. In India, there's insect-resistant Bt cotton that has been used for close to 20 years by now. In Bangladesh, there's Bt eggplant, which has already also been used by for 8 years now. And there are studies that have really used very detailed data independent studies. We have done research on India for many years. This is coming from a different research group, showing that the introduction and the adoption of these technologies by smallholder farmers has reduced chemical pesticide use, has reduced poisoning incidences because the small-scale farmers spray pesticides with backpack sprayers and without protective clothing. So there are poisoning incidences, and these are reduced through the technology, higher yields, much higher incomes. And for India, we have also analyzed over a number of years food security effects. And Bt cotton does improve food security, not because the cotton is so nutritious, but because this is the main income source for many of the smallholders. And when their income grows, then they can afford better foods and better diets. So one can certainly ask the question, what does that mean for Europe? Do we need GMOs for food security in Europe? Well, the answer is no. And the reason is because we are rich and we can buy whatever we want. When we don't produce it ourselves, we can buy it from elsewhere. And that's exactly what we do. In Europe, we import a lot of food and feed from other parts of the world, but this contributes to scarcity and environmental problems elsewhere. A recent report by WWF showed that European imports actually account for 16% of the tropical deforestation. And we all know that this is one of the huge environmental and climate and biodiversity problems, deforestation in tropical areas. But when we import soybean meal, when we import palm oil, when we import meat from Brazil, this is what contributes to land use change and deforestation in these regions. We have done very recently a study where we have looked at what would be the greenhouse gas emission effects if the European Union, this is referring to the European Union countries, would adopt GMOs, namely those GMOs that are already available and already being used elsewhere in the world. And we calculated that this would reduce greenhouse gas emissions worldwide by 33 million tons of carbon dioxide equivalents per year, which is equivalent to Europe's 8% of Europe's total greenhouse gas emissions from agriculture. So this is a big effect, and it shows clearly that what we do with our agriculture in Europe also matters. And this is only referring to those types of GMOs that have already been commercialized somewhere, maize, canola. A lot of the things that could be of more interest for Europe haven't even been commercialized because Europe is so reserved against this technology that things haven't been brought to market. So the effect could actually be much bigger if we would allow technologies that are more useful for Europe. Some of the major issues with Europe's GMO regulatory approach, I've already mentioned that it ignores the state of research because there's no scientific reason why GMOs or gene editing would be more dangerous than any other crop technology. But it also fuels public fears. I mean, the public obviously thinks if these are so strictly regulated, something must be wrong with this technology. So there's the politicians who are saying, well, the public doesn't want, and the public saying, well, if it's so strictly regulated. So it's mutually reinforcing fears and misunderstandings of the technology. It's basically a ban on very useful technologies for sustainable farming in Europe, but it also has major implications for other regions of the world. For instance, countries in Africa and Asia are looking at what are Europeans doing if they are not, if they in their high-tech and highly intelligent populations are not accepting these technologies, something must be wrong. Countries also feeling that we cannot move forward with these technologies because this would jeopardize our export markets if you want to export food and feed to Europe. In the end, we need a new discourse, a more science-based and more evidence-based discourse about new breeding technologies rather than prejudice and misbelief. And I hope that this webinar can make a little step into this direction. Thank you very much for your attention.
Thank you very much, Matin, for what has been for me a very clear and concise talk. Are there any questions of understanding? So I would wait for those if there are any. And thanks for posting already quite a few questions for the Q&A session. They look great and I'm looking forward to that. But let me just check if there's a question of understanding coming in. No, I don't see any. So I'm really looking forward first to the third speaker, which is Angela Bearth, who is a senior researcher at the Consumer Behavior Group of ETH Zurich. She is a trained psychologist and she focuses on issues such as how the public perceives scientific uncertainty of new technologies like gene editing, food irradiation, and pesticides. She talks about how consumers view and think about agriculture and plant breeding.
Thanks a lot, Thomas, for introducing me. I'm happy to talk a bit about the consumer perception and feel honored to be able to do that after my previous speakers. Consumers' perspective of agriculture and plant breeding, and specifically biotechnology, is a very large topic. So you could spend a lot of time talking about it. A lot has been published, a lot has been discussed, and I would like to focus on a specific topic today, which is how do we assess people's opinions? Apologies for our English-speaking audience. I have taken this study from the Bundesamt für Statistik, which has asked people over the years what kind of dangers they see for humans and environment for the variety of topics included in this excerpt from the larger figure. So for example, they asked them, how dangerous do you perceive loss of biodiversity? How dangerous do you perceive pesticides? Climate change, nuclear energy, and among them also gene technology. And if we look at the data, we can see that people perceive gene technology to produce food similarly dangerous as other topics such as pesticide and climate change. The question here is that the right question to ask? Because it only focuses on risk perception. While risk perception, of course, has implications for acceptance of a new technology, it's very limited because it doesn't include any of the potential benefits it might have. It doesn't explain much in terms of trade-offs that we might want to do. Similarly, also a rather simple way of asking people what they prefer or what kind of acceptance they have would be to ask, would you prefer to have a normal potato or a genetically modified potato, where probably a large number of people would say, I prefer the normal potato. This because we have heard scary things. We have read scary things on the internet about genetic modification. It sounds unnatural, it sounds dangerous. And I believe this is a sort of reductionist way of asking consumers about their opinion on these very complex issues. I would like to spend a bit of time to think about how consumers make decisions and how they think about complex scientific issues. First of all, we have an area where science is pretty sure about, where science knows sort of the role that a certain technology could play. They know how it works, how it can be improved in the future. And then there's, of course, also some uncertainty. So things that we don't know yet or that we don't know. This is the area where experts are in. So an expert has a lot of knowledge about a very specific topic or about a technology, whereas non-experts, of course, we don't have time to spend to also become expert in every single topic, in every single complex issue. So non-experts usually have to rely on less their knowledge and more experiences they made. Or for example, when you've read something on their beliefs and values, but also so-called heuristics and biases. Those are short rules of thumb that help us make decisions. This is very useful in general, because if we would have to weigh the pros and cons of every decision every day, that would make it very hard for us to take decisions at all. So this is helpful for us, but for complex issues, this can sometimes lead to suboptimal decisions for ourselves, but also for society. We know from the vast literature on consumer acceptance of biotechnology that we mostly base our decisions on three types of heuristics. So first of all, the affect heuristic, which says that we base our decision-making, our judgments on the feelings evoked by a certain technology. So if we hear gene technology, we have certain associations based on what we've seen, based on what we read, and we might have a positive or a negative feeling regarding this. And then we take our feelings as a sort of pathway to a decision. So when we have negative associations, negative feelings about biotechnology, this would lead to lower acceptance, or we would perceive it as something risky. Similarly, we rely on trust in the absence of knowledge. So if we don't know ourselves to judge something, we rely on the trust we have in people that do make decisions. That is something we rely on. And the third heuristic is the natural is better heuristic, which means that we perceive things that we think are natural as better. So safer, less risky, healthier, and so on. And these three heuristics contribute to responses that we give to very simple questions. We've heard before, we don't just want to use genome editing because we can. We want to use it to tackle certain challenges. And so in our study that I want to briefly discuss, we decided to first give the people a sort of understanding of what kind of issues we're facing. One of the issues in agriculture is potato blight. We've heard it before. This can rapidly spread in potatoes and can lead to losses in potato crops. There exist a number of solutions for potato blight. And we presented to people with 4 different solutions. We spread the participants in the study into 4 groups. The first group was presented with fungicides as a solution for potato blight, which is normally used in conventional farming. The second group received the information that in organic farming, copper is sprayed on the potatoes to make them resistant. And the third group received the information that genes of a wild potato that is resistant to potato blight are transferred into the cultivated potato. The last group, we introduced a solution of gene editing, so changing some information on locations in the genome of the potato to make it resistant. So we presented people with a certain challenge that agriculture faces, and we gave them different potential solutions. I'm sure you're curious now to see the results. Here we asked people about their acceptance and willingness to eat. On the left-hand side, you can see 0 corresponds to a low acceptance, 100 corresponds to a high acceptance. Interestingly enough, in our study, the gene transfer, which corresponds to gene technology, exhibited the highest levels of acceptance, whereas gene editing, natural and synthetic pesticides were equally high in acceptance. We can also see that the acceptance is not as low as we would expect based on what we've heard before. So it's above average, the mean of the respondents. I've talked before that naturalness perception plays a role. So we also asked people how high is the decrease in naturalness after this treatment. And what we can see is the highest decrease was seen for synthetic pesticides, whereas the decrease was lower for the other solutions. Now, there's not a lot of research out there yet that focuses on differences in perception of gene technology and genome editing, but we did a study on tomatoes where we presented the same people with information on gene technology, but also on genome editing. And then afterwards, we asked them, how similar do you perceive these technologies? And what we can see is that roughly 50%, so half of the participants, perceived these 2 technologies as different. Of course, there's also some people that perceive them as somewhat neutrally, and then there's some people that perceive them as not different at all. But the majority sees some differences in these 2 technologies. As I've stated before, there's a scarcity in the scientific literature at the moment that focuses specifically on genome editing. However, what we know already is that there's some positive attitudes towards genome edited food, particularly in younger generations. It's seen in a more positive light compared to other older genetic modification technologies. And what we also see is that benefit perception is an important factor. Of course, we all would like to have a normal potato if we are asked in this very simple way, but our world is challenging and we're faced with increasingly more challenges, and so we should make use of certain benefits. Here are the takeaways I would like you to take away with after my presentation today. In the absence of knowledge, consumers use heuristics to make decisions about complex technologies. Makes sense, we don't have time to become experts everywhere. But the type of questions that we ask consumers to afterwards decide whether people want something or not matter. So if those questions are oversimplified, we're going to get simple answers that probably don't reflect the true meaning that consumers have. Also important, the older consumer studies on gene technology might not be as informative on consumers' views of genome editing. Thanks for your attention, and I'm looking forward to the discussion.
Thanks a lot, Angela, for another nice talk. If there are any questions of understanding, we are waiting for those. And if none are forthcoming, I'm really looking forward to an animated Q&A session. We have already 11 questions that came in. I'm still waiting a little bit for question of understanding. So that doesn't seem to be the case. Another case of a very clear talk, not needing any such questions. So let's start with the Q&A session. Let me remind you, you can add your question in the Q&A box. You can upvote questions if you like to put them on top of the list. I started with one that was upvoted, and I guess that's probably for Dirk, but whoever wants to join in, then of course, please feel free. The question is, what is your opinion on de novo domestication?
Well, I think it's an extremely good question. I think in fact, we know from work in a number of crops, for example, in maize, that in fact, there are a number of major genes which have been contributed to domestication. And in recent examples, particularly in tomato, it has been shown that you can rapidly convert, let's say, a relatively wild tomato variety in something which looks like a normal high-yielding tomato variety. It's been published 2 years ago by a group in Cold Spring Harbor. And the additional advantage was that this tomato produced something like 5 times more lycopene, that is the health promoting substance, the red color essentially, which is in tomato. Well, in fact, so you rapidly can domesticate relatively wild species, wild low producing varieties in a very fast way, and furthermore improving their characteristics. So this is enormous perspective for crops which are now underused because they are not very well adapted, they don't produce very much, to rapidly to breed them so that they become more productive. And maybe by increasing the variety of crops we can use like for European agriculture.
Let me maybe ask a clarifying question. If we are talking about this de novo domestication, can this be done by any kind of technology, or is it really only with CRISPR-Cas that you then are able to do it so fast?
With breeding, because you need so many changes, like it would be virtually impossible to achieve all of that, because it would just be too time consuming. And because we have the knowledge now, really to on the genes which needs to be modified in order to domesticate. And this in the final end product, there will be no foreign DNA anymore present out there. So it's a matter of changing the existing some of the, let's say, what is called expression. So the strength of the existing genes which are present in the plant genomes, and we know that, for example, in maize, everybody knows in maize plants, this is one stem, and then you have your cobs growing into that. Well, we know that that is due to a mutation in what is called a gene called teosint. And that mutation, the original maize had many branches. I mean, many, and so that single mutation actually changed that the many branches to one stalk, one stem essentially, and that was a great improvement, like in the production, the development of the modern maize varieties. Yeah. So it's a breeding method essentially. Like that's essentially a way of introducing genetic variation. But now in a much faster way, and that's what Matin said. I mean, I think we really, I think it's important that, I mean, the precautionary principle, in my opinion, is important, but we should also invert this question. What if we do not have access to this technology? I mean, there are so many fantastic things which can be done for the planet in terms of climate change, biodiversity, and so on. And it would be a shame not to use this technology for in the European continent because of fear mongering and people are uncertainty that people have about all that. Yeah.
Let me add here a question that that has been that kind of fits to this, that has been asked in the Q&A. Somebody says also in the first kind of generation of gene technology, namely the classical one, you know, there were lots and lots of promises made, he says, and or she, I don't actually know, and some of them apparently were not didn't materialize. So what's different this time? You know, when are there any products, the question is, on the market that really are useful for consumers? And then this person asks, and maybe acceptance will also be bigger.
Yeah, that's certainly true. I mean, an enormous amount of promises were made at the beginning as saving the world like from hunger and all these things. And this technology is different because the ease by which it can be applied. It's in fact, it's very simple to do that. I mean, of course, you need a laboratory for that, but relatively simple to that. Furthermore, in fact, this. Progress has been made in our enormous progress in our understanding of what makes a crop today. So we have now enormous advances in genome sequencing. We determine genetic information of crops, and we have learned what we have been breeding for. And we know it's not a single gene. We know it's actually, you need combinations of genes. It's like breeding in order to get your favorite trait. And that's where genetic engineering, the classical genetic engineering failed, in fact, because we're always talking about a BT gene or an herbicide resistance gene. It's a single gene. Like if you want to tackle a problem like drought resilience, then it becomes much more difficult because you need multiple genes, and that's virtually not possible to approach by GMO technology. At least still much more difficult to do that. And furthermore, the legislation made it so difficult to bring new products on the market. It's been estimated that you need $100 million or euro to bring a new GM crop on the market, not in the European market, but doesn't work, but in the US or South America. And so it's enormous. The hurdles are so high that only the big companies can in fact put that money on the table to achieve that. And so that's something that we really should avoid in Europe. As I said, you cannot distinguish what you obtain with genome editing from normal breeding. So it's actually ridiculous that you would regulate something on the way that you achieve it, if the end product is the same. You see, it's like, I mean, I give you an example. You have a complicated text, like a book, and you know that there is somewhere a word which is wrongly spelled. and you want to correct, this is the genome, you want to correct that word. You can read this. The old method is to read the book from the beginning to the end, and you find that word. The modern method would be to take your computer and find and correct like a command, and you have it immediately. That's more or less comparable.
Okay,
so and
thanks.
the end product is a change in that thing, and you so difficult to see how you can regulate that and stop this technology. Sorry, I said enough.
No problem. Thanks. So Martin, I think you also wanted to add to that.
I'd just like to further emphasize one point that Dirk already made. I mean, of course, there are technological advances, but indeed, it is true that many of the things that have been developed, even with the classical GMOs, have been held up in the regulatory thicket. There are so many interesting types of applications that have been tested around the world in field trials, including field trials in countries of Africa concerning drought tolerance and nitrogen use efficiency. But they were never approved. They were held up for years and years, and no African regulator and policymaker dared to stand up and say, look, we move this forward because looking at Europe, that cannot be a good idea. So, of course, I mean, gene editing has lots of new potentials, but what really killed the transgenic GMOs is the inefficient regulation that has made it extremely costly, but that has also made it extremely lengthy. And you can't even see lengthy. If there's never anything approved, then it's an infinite period. And of course, it becomes a self-fulfilling prophecy that all those promises remain unfulfilled. That's not to say that there haven't been overblown promises back in the 80s. There were, for sure. But a lot of the things or some of the things at least that were promised are already there, only that they have never been approved.
Well, let me add a question here that, Angela kind of touched very closely in her talk. That she was saying that people kind of are probably much more willing to accept such a thing if they have a personal gain from it. And I mean, what people usually talk about is really herbicide resistance, for instance, as Dirk has shown, and this all kind of agricultural system that comes with it, and all the discussions we had in the last years. So my question is, why did all these plants pass through the legislation and all the others, which kind of would have had benefits to people, but also to nature, which I think might have the same effect for people's acceptance? Why didn't they pass?
I mean, you started your question with Angela, so I'm not sure whether you were
no.
particularly looking at
think
Angela.
I mean, whoever. I think Martin, it's you're a good point, but then Angela, of course, as well.
I can keep it brief, and then Angela can certainly add to that. Well, if you look at herbicide tolerance, when this was first commercially approved in the US and then soon later in Latin America, that was in the mid-1990s. The same holds true for BT. These are the old technologies that were approved actually before the European Union had come up with its new regulations, and before actually the protracted discussion started. And this is exactly the reason why we are still looking at these, you can almost say, outdated technologies that are 25 years old, and nothing has really happened since then in farmers' fields. It has happened in the labs, it has happened in the field trials, but not in farmers' fields. So the last thing that was approved for commercial use in the European Union was a BT maize event by Monsanto in 1998. 1998. Since then, there were many technologies that inventors applied for getting them approved. They were not, and not because they're dangerous. EFSA made always its opinion. I mean, EFSA is the European Food Safety Authority that looks into the risk assessment documents, has made repeatedly opinions that this is safe technology, but the politicians never moved forward and approved this. So this is the story behind how Europe has dealt with GMOs. And we are currently running the risk of making that same huge mistake for gene editing, and that's the real tragedy about that. Sorry for being too long, Angela.
No, not at all, Angela.
No, you were the perfect person for that question, I suppose. I wanted to, it goes a bit in a different direction, but I wanted to talk a bit about the perception that experts have of the public when it comes to this topic, which I've also read in some of the questions, is the perception that people really don't understand, and they really don't want to, and they don't believe us when we say it's safe. They don't believe us when we say it has certain benefits. And I think this might be a sort of misconception on the expert side. So I think when it comes to scientific topics, of course, there's always debate, public debate, there's misunderstandings. But in general, I think the majority of the people are just uncertain. And then they're discouraged by certain things they hear. But I really think if we discuss more frequently about we need to do this, sort of also the other perspective that Dirk mentioned, what happens if we don't do that? What happens if we have climate change and droughts? And this is a technology that could tackle that. And I think it would be good for experts to also focus a bit on just the uncertainty that people feel instead of the perhaps very loud minority that is very strongly opposed against anything that seems unnatural.
There is a question which got the most votes that really follows exactly to what you are saying now, and it says when 30 years of risk research has shown that GMO plants pose no risk, so why didn't this result, if you like, kind of end up in people's heads already? How can the benefits be communicated better and more easily accessible for people? I guess, Angela, maybe first again, and then whoever wants to chip in, because I think that's really a key question.
I think, as I mentioned before, we're busy people. We have a life, and we spend our times, we're all experts in something, be it our hobby or our jobs. So we don't have time to follow this in detail. Not everyone has time to follow this in detail. So that's why we make use of heuristics. That's why we rely on something that we've maybe heard years ago about something. And then this is the association we have, and this is what we stick to. And it's very difficult to add new knowledge about it if it's not that interesting to us, or if we don't have that much time to actually spend on it. So I think even if benefits appear, they're much more difficult to sort of get the word out.
Martin?
I mean, when we look back 30 years, even scientists at that time had much less knowledge about potential risks. 30 years back, there was not a solid statement that one could have made that there aren't any new types of risks. And this is when the debate started. Since then, we have 30 years of additional research. We have basically all the academies in the world that came up with scientific consensus statements about not that there are zero risks, because there are risks with any technology, but the statement that the risks do not systematically differ with what we have done before. And that's something that hasn't made it really sufficiently into the public debate because people don't change deeply rooted beliefs that they still have from the debate. That's refreshed by statements of anti-biotech interest groups that against better knowledge are perpetuating, half-truth and risk and horror narratives. And this is perpetuated by the media that tries to be balanced. The media often considers balanced that everybody gets the same voice. Fortunately, in climate science, we are beyond that. I mean, there are still people who are doubting that we have climate change or anthropogenic climate change, but that's such a small minority. And fortunately, the media doesn't give them so much room anymore. But that's not the case for the risks of new breeding technologies, where we have a very broad scientific consensus. And of course, there are a few who don't go with this consensus, a few, which is 1% of perhaps, but the media gives them the same weight, and that certainly perpetuates the beliefs that, well, if you always hear about those things in various forms, they must be true. And then comes Angela with a very correct statement. People are not experts. They don't invest huge amounts of time in order to update what they have once, declared as this is risky, this is evil. And that can lead to such strange situations that you have a huge majority of scientists and scientific consensus statements, but it has relatively little effect on public beliefs.
Dirk?
Yeah, I would like to add something to that discussion. Like, I think the picture of let's say the old GMOs, the BT and the herbicide resistance is also associated with an agriculture system, which is maybe very different from ours. It's a large scale mega farms, like all. If you go to Iowa, like all the same cornfields as for hours to drive with a car, like and so it is also associated with certain companies, and not even the name, like with the kind of exploiting the nature and so on. Like while in fact, like this picture has been amplified, like by certain groups, like, and at some point we can even I think they might be right actually to go against this kind of model for agriculture production. Now, in fact, with the genome editing, we have a chance maybe to adapt our agriculture to the European way of farming, more smaller scale, maybe more integrated in the landscape, maybe more eco-friendly, more variety, so that we have this typical taste which we are also used to it, that we can make a new agriculture system. So that is the opportunity, in fact, with this new technology, because it's so easy to apply and to change the varieties. I think at the same time, like in fact, like we as European, and Martin has been saying that very clearly. I mean, we have been exporting our problem to other parts of the world. Like in fact, it's really, I mean, we are responsible as European, like, for the deforestation, like, of large part of the Amazonian forest, because we import GMO soybean because we cannot grow, we cannot even improve protein crops, like here. So again, this is a possibility to really make our agriculture production system more modern, more adapted to our way of living, and in the same time being eco-friendly and saving into some aspects also the climate and the biodiversity of our planet. It's, I think, really an enormous opportunity that we have now.
Let me pick up and add a question that was posed, which fits to here as well. You mentioned it also in the talk that this technology is relatively easy to use and so cheaper and more accessible to also small companies. And there's a couple of questions related to that. If that really is the case, I mean, one of them says, what is about the patent situation? Is it really still easy for small companies to use CRISPR and then to patent this plant you made with it? So you just CRISPRing something in the lab might be easy and cheap, but then the whole thing behind it is maybe much more expensive, and you need a big cloud to actually do that.
Yeah, that's indeed a very good point, and the patent situation is not entirely being cleared. It's presumably the most patented area of biotechnology in the last, maybe together with all kind of variants of vaccines, I guess, and of biotechnology the last like a couple of years. But I must say that this technology, this CRISPR technology, Cas technology, comes in many different flavors. It's not only one system. There are many variants, and now we have already a number of different companies which are really have various let us say variants like of the CRISPR-Cas, not to be too technical, which really are competing to each other. So we see now that in Argentina, for example, where there are already several CRISPR genome editing crops entering the market, like really that there are really no major hurdles actually to do that. Because if some company actually prohibits the use of a certain tool, like in fact there are other companies which can ship in and provide it. So this competition in that area, and that I think is a very good point. But so this I think it makes the hurdles like much. We have these hurdles, but they are certainly reduced. Like in fact,
Martin, I think with your experience, you might chip in here as well.
Yeah, I think the the question of patents and other types of intellectual property rights is really crucial because we know that inefficient patent management can lead to industry concentration. We know that from research over longer periods of time. And this is something we have to keep in mind. But this is not an argument against or for a technology because the technology is not coming inherently with patents. Policymakers can certainly decide what is patented and what can be patented and what not, whether you can patent varieties developed with gene editing or not, whether you can develop certain knowledge and enabling technologies. And this is something where we need a serious debate about, but the debate is not about whether we should allow gene editing. The debate is about how would we efficiently manage gene editing and possible intellectual property rights around that, such that we can ensure competition and diversity in the seed markets. A very important point needs to be differentiated a little bit from a black and white discussion about the technology. And I'm not saying that we should leave that out here, not at all. But we should not say we don't want gene editing because we don't like patents on plant varieties. I personally don't like patents on plant varieties as well, but that's an important statement that comes with the question of how to politically deal with this.
Okay, so let's move on a little bit to an interesting question that ties to something that was said in the talk as well about Chile, which went ahead with this technology. And the question is, will EU seed companies still be allowed to do counter seasons in Chile? I had to ask back with our technical people what that actually means. It means apparently that they can grow crops in Chile in off-season Europe and sell these. So would that still be possible if for these companies from the EU, as it's not allowed here, this technology, or if their crop is not legal here? Yeah, and can you actually then check these editing events in the plants if they are reintroduced?
Yeah, so actually breeding companies, they make all the time use like of the difference between the southern hemisphere and the northern hemisphere in terms of climate. And like in fact, so it's a very common practice that a breeding company do a field test like here and in summer, and then do another field test and in our winter, then in Argentina, Chile, for example, and so on. And so with the current legislation of genome editing, that would not be possible anymore. Like, so that means where many of these companies now global players, like in fact, it would now, if you have a genome editing crop developed in Chile and you do a field trial, and you would like to do that to continue with further improving it by bringing the modified the improved characteristics in other varieties, and you would like to do that in Europe, it would not be possible anymore, because it would be considered as you import a GMO, even when it is one nucleotide, one of these 3 billion letters in the genetic code, which would be modified. Even one that would be sufficient to consider it as GMO. You cannot detect it. You can only detect it when you know it, that it is there. So when somebody says you, okay, this variation has been created by genome editing, then you can detect it. But if you don't say it, if you don't add this information, it's impossible to detect it, whether something has been made by genome editing or by conventional breeding. That makes it also very difficult to trace. That is totally different from a classical GMO. A classical GMO, you have a foreign piece of DNA. In this case, BT, insect resistance is from a bacteria. You can detect it, and you can detect it even when it is 1% present, in an imported food, for example. That's going to be much more difficult for genome editing crops. Dus in practice, it creates enormous problems for breeding companies. It's putting Europe in a very in a situation where it cannot be competitive anymore in terms of breeding, because you cannot exchange material.
Okay, thanks. So next question is, what are potential pitfalls on crop breeding that would mainly rely on CRISPR gene editing techniques? So are there special things that might happen there? I mean, I also, when I read reviews before this, you know, they were talking about a lot of hurdles and problems it's not as easy that you just get whatever you want, but there are some off-area introductions and things like that. So that would be an interesting question, I guess.
Yeah, I can answer that. So we are talking about what is called off-target changes in the genome. So genome editing, it's a method to really to do something very localized and predetermined site in the genetic information you bring in a change. But in the earlier days when this technology was developed, the first experiments were done on mammalian cells. That's where, in fact, let's say this 4 or 5 years ago, so they have been doing that. They have been sequencing the entire genome of these modified cells, and they found that next to the site where you want to have where you want to make the change, there were also, with a low efficiency, also some changes here and there, in the genome. And so that was immediately, some people called this, this is uncontrollable off-target. Well, it turns out that is really specific for mammalian cells. This off-target mutations are very rare into plants. They occur, but they are very rare, in fact, and now with the improved gene editing, almost they are completely absent. So people have been sequencing genomes obtained after genome editing, and they do not find off-target mutations anymore. And and furthermore, one should not forget that the mutations that we look for with genome editing is very small mutation at one particular position. If you do mutation breeding, or if you do even classical breeding, where you mix plant genomes by crossings, or you irradiate plants with mutagens or with X-rays, and to make variants, you introduce thousands of mutations all over the genome. And nevertheless, this has never been considered as something dangerous. It's actually that's what we are doing. If that's what biology is. people have man and wife have child, they inherited genetic information like from you and your partner, like, and you yourself have genetic information of your grandparents. There's a complete mix of genetic information. That's what breeding is doing. And now what genome editing is actually doing is that really making specific changes and like and repairing things. And now there are a number of diseases and like in humans and heritable diseases, which like sickle cell anemia and so, which can be efficiently repaired like by this technology. That's a complete revolution in that area. Sorry, Martin.
Yeah, please.
No, just one thing, because I think the question on potential pitfalls of over-relying on gene editing has another nuance, namely the nuance, can we use gene editing to substitute for other types of breeding? And I think the answer is a clear no. I mean, it's a very efficient and precise addition in the toolbox, but it's certainly not substituting, at least for things such as crossbreeding and selection in a local context, for developing locally adjusted germplasm. And then you can use gene editing to add interesting traits and characteristics. So if that was perhaps also implied by the question, I think we should not, at least as of now, believe that gene editing will be the only tool that will be used by breeders in the future. It won't. It's just a very powerful addition to the toolbox.
I mean, can you at the moment already say that you can change whatever little small piece of DNA in whatever plant you like precisely as you like, or are there still hurdles to do that?
There are still hurdles in the sense that you rely on some laboratory technology that to what is called to regenerate plants. In fact, what you're doing is to take a little piece of a plant and you temporarily introduce this CRISPR system, makes the mutation, but then you from that little piece you still have to regenerate, to recreate a plant. So it's and that is for some species rather difficult to do. So that's the technical hurdle, but I mean also there enormous progress has been made, and I think Martin made a very good remark in the sense it's still part of a breeding program, and so it's actually creating genetic variation at a locus where you want it to be created, because you know that it is important. It makes wheat, for example, sensitive to this fungal disease. Like, so you want to create their genetic variation. But then this, once you have introduced this mutation, then this plant enters a breeding schedule. It will be crossed with all possible varieties to bring that characteristic in these varieties. They will be tested in the field to really test whether they increase the resistance to that fungus. And only the lines, like in fact the improved varieties which do it in the field, those will actually be submitted to regulatory commissions, which really will compare it in comparative test with existing varieties. And only when that new variety, that CRISPR obtained by CRISPR, when this variety is doing better than outperforms the existing varieties, then it will be given a permission to be introduced in the market. So it's part of a breeding cycle. It's just a way to create genetic information, genetic variation, but now very targeted. And so that's important to realize. And I mean, and one can think about all kind of doom scenarios, but I mean, there is a whole food safety legislation in Europe which is very strict, and which really controls like all the possible steps into that, that which safeguard that the product which comes on the market is actually safe.
Okay, thanks. So let's move on to the next question, which I think is for Angela. What is the influence of the organic lobby on the perception by the consumers?
Well, I think lobbies definitely have an impact, because the way we're communicating about things, you have certain goals with the communication you do, so that definitely impacts. So any sort of newspaper we read, any sort of information we read that has an impact. But I think that's maybe not the most important issue. I think the important issue is to talk about how regulation and policy makes decisions, because that's going to be much more impactful. And as I said before, we can of course achieve certain things if we explain to people. Knowledge provision has been shown to change opinions and to increase acceptance, reduce risk perceptions, but that's not gonna lead to every consumer going, yes, I love that, which is probably also not the goal that we want. But if we ask them about their opinions, because we agree that we want to have a democratic process and we want to include the consumer in these decisions about technologies, then we need to make sure that we take them seriously, and we don't just ask them, do you think it's dangerous, and then just ignore everything else. So I think that is maybe the topic, that we need data on consumer acceptance that then policy and politicians also can use, that really reflect people's informed opinions. So I guess lobbies do have an impact, but I'm not sure whether this is something we should focus on too much.
But that means, in a way, I mean, just to get a better understanding, what you're saying is that in a way one should start asking more and more nuanced questions to people.
I think so, especially if this data is then used to inform policy and decision making in the political decision making. Then we need to make sure that we indeed ask the people the questions and get their correct responses. And I think this is currently not really done. We pay a lot of attention that the data is representative, which is of course important. We get large data sets, but we ask people very simple questions, and then we judge from these simple questions or from the simple responses that we get, we judge that people would reject this technology, which I think should be tackled.
There is one question which kind of ties into that as well. Christian Funkhauser makes the link to COVID-19 pandemic, where he says that even though it's shown that that science is actually helping a lot with vaccine development and other things, still many people do not want to accept scientific evidence, and the question is, do you have any advice on how we can try to convince the public, even if things are apparently really clear?
Well, I think I would disagree that we really need to convince the public that much of the vaccine. I know there's studies out there, but similarly, they were very large-scale studies asking very simple questions about vaccine hesitancy. So the Tagesanzeiger, for instance, asked people in October whether they would get vaccinated. And I wasn't surprised that many people said no, because back then Swissmedic has not approved any of the vaccines yet. So people were thinking of a hypothetical situation, of a hypothetical vaccine that hasn't been approved yet. And so I'm not surprised that people said, I wouldn't at the moment. I think if we ask people the correct questions, then they would be much more willing to get the vaccine. So I think perhaps there's a bit of an overestimation of vaccine hesitancy here in Switzerland. There's also data that suggests that, for example, trust in science went up during the COVID crisis, which is, I suppose, hopeful and sort of rejects this idea of everyone wouldn't want to get a vaccine and everything's a bit hopeless. But I think for those that have vaccine hesitancy, there's definitely a toolbox from the social science on how to improve this. So sort of one-on-one talking to people, taking their concerns seriously, because it's something uncertain that comes towards us. So we kind of want to take their concerns seriously. And this, I think, transfers nicely to gene editing. So if people have concerns, perhaps we need to talk about these specific concerns.
It's actually a really nice question that just popped up here on top of on the second part of the list, which, it says, at this rate, who should be persuaded first, the government or the population? Which one would be more impactful for the acceptance of GMOs? So that's in a way the question.
I'm really interested in the responses from the other two, but just a quick response from me. I think the politicians should be persuaded first.
Here I would disagree, simply because I'm talking to a lot of policymakers from all parties, including the Green parties, and many know what the science says, but they know that behind closed doors. And many say, but look, I mean, in a democratic society, what can we do? We're not going to do hugely unpopular things if the majority out there doesn't want this technology. So I think, of course, I don't really disagree with you that talking to policymakers and governments is important. But I think in democratic societies, which is great that we have them, and it's great that we have a lively and dynamic civil society, but we cannot move forward with things without having a serious science-based public dialogue. And I think that's something that we had to learn. And it's also obviously not sufficient if a few researchers say this is good. That's not working that way, Angela told us, and we need to find better ways. But we're not succeeding if we only talk to the governments because many of them know, but what can they do against public will?
I tend to agree with that view, Alec, and I think what is the more important question is, what do we want as a society, Alec, and with our agriculture production, Alec? I think it's very convincing that in fact some of the goals of organic farming, I mean the prohibition of chemicals, is also among the top priority of people who think genome editing should be applied. So there's actually common goals. Also, actually, maybe increasing the amount of carbon in the soil by having plants with deeper root systems and all the system and so on. So we can, I think, many of the goals are in common. And one has to ask the question: do you really want to treat your diseased plants with copper sulfate, or do you want to have a plant which have a built-in resistance? I would personally choose for the built-in resistance, because copper is absolutely not healthy for you, as well. And although it's a natural, it's not a synthetic molecule. So we really have to ask. I mean, organic farming is often and it certainly is a role in society. In fact, in many areas in Europe, in fact, you don't want to intensively farm. I think in Switzerland you have many of these meadows. I mean, you just leave it like that. It's fantastic biodiversity. But on the other hand, really to propagate that, you need everywhere in Europe organic farming. You have to accept also that you need much more land. It's just much less productive. In fact, it's been quite a large number of studies show that you need approximately 50% more land to produce the same amount of food. In fact, and so where is this going to? Are we going to destroy forests, or to have more land, or going to again increase our import of foods from South America? So it's really an important decision that we have to take. So I think there is room for both models, but they rather should use the benefits of both sides rather than trying. Now there seem to be a kind of you choose for the one or the other, but there is, I think, something in the middle which is maybe much better for Europe. In fact, it's so that's the debate I think that we should have.
But then doesn't it mean, in a way, when you're saying, that there are many links between what people working with CRISPR-Cas on plants and what like the organic movement wants, that there should be a much more closer link and try to kind of talk and work together to come to an agreement what would be something that is one should target in terms of how you go ahead and improve plants.
Absolutely. I mean, I think the scientific community at large is a big advocate of this kind of dialogue. What do you want as a society? And what do we understand under sustainability? What do we understand eco-friendly agriculture? What is it for something? What is organic? What does it mean? And so we need to have this discussion, I think, and really look for what is the best approach, like in fact, to in the same time to preserve biodiversity, but also the typical European landscape agriculture system, and at the same time also food security, like in case that we have again a serious drought, like that we have sufficient food to feed our people, like. And so this is the kind of debate which we urgently need, I think, like and not trying to get this kind of war. It's a fight between organic and the gene technology. Like there's something in the middle which is really worthwhile to explore.
There's one kind of critical statement and question is that that with classical GMO, there's lots of instances where plants which had traits which might be resistant against certain disease or pest or whatever, that then these diseases or pests develop resistance against those, like BT, I think it happens, and so on. So isn't the danger that with many of these, also with the newer kind of generation plants, that happens, and shouldn't that be kind of a way how to that one has to take that in account if one also works with these newer things, and that it's not just kind of a new solution which gets rid of all the old problems of the old plants, as you called them as well. Yeah, Martin?
I mean, the resistance development against pest control agents is something that we know exists with any type of pest control methods, whether it's a biological pesticides, whether it's a chemical pesticide, or whether it's a genetically inbuilt resistance mechanism. And we need to be aware of the fact that evolution happens and pest populations have a tendency, of course, to evolve in ways that they can still be overcoming any resistance mechanism or any chemical pesticide. And that's a matter of fact. Of course, we would be ill-advised if we are not taking that into account. I mean, this is not different for gene editing or GMOs or chemical pesticides or biological pesticides. The only thing we can do is try to manage resistance development in proper ways. That's a question of proper agronomy, using systems that help reduce this. And of course, we are also ill-advised to say now we have a resistance mechanism and we plant monocultures because this is leading up to increasing pest problems and resistance problems. We need diversity. We shouldn't use new breeding methods to overcome diversity. The contrary is true. We need diverse systems. We need to combine proper agronomy with breeding and other types of technologies. The good thing is that we will likely see increasing pest pressure problems in regions that we haven't seen so far with climate change and global environmental change. And gene editing offers one tool of being able to relatively quickly react. This is great, but we cannot jeopardize that by saying, well, we throw good agricultural practice and agronomy overboard. That would be a bad advice. So I agree with there are certain things that we need to keep in mind, but that's nothing specific to gene editing.
There's one question which is kind of specific now for Switzerland. It says, assuming that Switzerland would allow GMO crop production, or I guess it's rather meant gene editing as the CRISPR-Cas system is called, would that enable an increased self-sufficiency, independence from import? That's kind of a topic that comes up in Switzerland from time to time. So basically, can we increase productivity of Swiss agriculture with gene editing?
Yeah, that's a difficult question to answer if one doesn't have any information on the specifics of the agriculture production, which is currently is the case like in Switzerland. I think it would allow to increase food, the more security in food production, maybe less danger of that you could lose crops due to inability to control pests, and I guess also to increase like productivity without increasing the footprint on our environment. So that I think I guess there are definitely a number of positive elements which could be added to the agriculture production in Switzerland. Whether you're going to be self-sufficient, I mean, I don't have and any of the data to really to calculate all that. Yeah.
But I'd perhaps also add that self-sufficiency, at least from a sustainability perspective, is not necessarily the goal. Trade is important to also, use those resources where they are and not overuse them where they're particularly scarce. But what's happening is if Switzerland and other parts of Europe, just think that switching to low external input agriculture, switching to organic would be good for sustainability. The answer is, well, let's be careful. It may have environmental benefits locally, but what we're doing is we're producing less, we are importing more, and we're exporting our environmental problems to other parts of the world, and that cannot be the solution. So I don't think that autarky and self-sufficiency should be the goal as such, but we cannot feel that we are an isolated island where whatever we do doesn't have effects on the rest of the world. We are living on one planet, and resources on our planet are scarce, and we should all see how we can use them in the most preserving way. But of course, we have to satisfy human needs, and I'm not that much worried about the human needs in Switzerland. I'm worried about the human needs elsewhere. Not because I don't like Switzerland, don't get me wrong. That was just a...
Yeah, well, it's an important important remark, I think. Like at if you look to the current GMO legislation, which really, I mean, Martin said already, which actually is being used also by many African countries, where they made it. I mean, now it's changing. Nigeria, so like and Kenya are also now having really the classical GMOs. So, but of course South Africa as well. But still, you see it's a kind of patchy pattern. Like in fact, like and exactly there. I mean, this technology could have enormous benefit and can. And certainly now with genome editing, which is actually a breeding tool, is actually very different from the classical GMOs. And like, in fact, we have a new chance, in fact, maybe to improve agriculture production in a more sustainable way in many of these countries where we know the big population boom will happen. I mean, now there are 1.5 or something like that Africans, and by 2050, 1.5 billion Africans. There will be 4 billion African citizens like by 2050, which is just 25 years from now, a matter of speaking. And so if we don't get the agriculture production system really up to a decent level, I mean, we're going to face a catastrophe, I think, in these countries. And so that's, I think, from a human point of view, it's so important to get this thing now right, that you allow modern technology to improve crops so that you can better varieties, less pesticides, because you anyhow they don't have money in many cases to apply it, like, or they, I mean, it's just so we have it's for the welfare of our planet. I think this is really important, and particularly for countries where they need it the most.
There is kind of a more of a comment, but kind of, well, let's make it into a question that ties into that, where it says, it has been lamented a lot about the kind of regulatory system of the EU, which you said is making a mistake in a way. And this person says, you know, when gene-edited plants can actually help with the real problems in agronomy, which, for instance, he says is, you know, having too much pressure on nature and on land and so on. So if they can contribute there, surely it will be authorized in the EU. So, I mean, what's your take on that? Do you think that will help?
Of course, but it's a vicious circle. It's you can. So if you, as an academic institute, if you want to do a field trial with a genome editing crop, so with one of the 2 billion letters which has changed, if you want to do that, you have to ask for permission. You have to fence your field. Like, you have to take all kind of precautions, and in fact there is there is a risk that one NGO detects this and comes to destroy the field, as we've seen like in France. Actually, they took the wrong field. It was a classical breeding field that they destroyed. Like it. So when you listen, even science is not given the opportunity to show that you can actually help for the environment. All the innovation will happen outside like Europe, and there will be, and there are already actually several examples like they're out there. So it's a kind of vicious circle. And I don't say that we have to completely deregulate all that. Maybe we can, as a society, we can say, look, we do not want herbicide-resistant crops because you have to apply too much chemicals. I mean, that's a decision as a society. That's a regulation. But now, like really on non-scientific basis, because you prohibit even the testing, the further develop of this crop, I think that is not right, and that's scientifically unsound, and it might be legally correct, but this is not a good thing for European citizens.
Maybe a question to Angela on that one. I mean, when Dirk is saying that maybe, you know, we should discuss as a society what kind of plant breeding we want for what kind of agronomy, should this not be taken up more actively by the scientists and saying exactly things like you just said, Dirk? I mean, let's not concentrate on herbicide resistance because this feeds into this really industrial kind of agronomy, which seems not to be what we want, and having this discussion really of very openly and very strongly with the public to get acceptance.
I think many scientists do that already. I have a lot of contact with different natural scientists, and I don't think there's better science communicators than people involved in biotechnology and gene technology, because they've learned the importance of talking to the public about their science. But I think it's also, many scientists do that in their free time because they have passion for their research and for their topic, and also experience a lot of backlash from extremist groups. I call them somewhat simplified. And so I think that cannot be the only solution. And it's also, we know knowledge provision helps, so it helps if we explain things, but it's not the whole solution. So it's much bigger, I suppose, because we are very far removed from agriculture. Not all of us, of course, here in Switzerland, but in general, we're removed from the whole process. So we have the luxury of imagining one single cow on a lonely field, and I think it would be important to perhaps talk as a public, talk about this picture of agriculture as a sort of lonely cow on one field, and discuss this, and maybe also discuss where the food came from before, so what we used before. That would help as a sort of tool for knowledge provision.
Okay, so we are nearing the end of this very interesting discussion. Let me switch gears for quite a degree. There's one question which ties to one of the slides in Dirk's talk. It's about wheat and gliadin and gluten response there, and the question is, in wheat, the gene crop methodology could reduce the gliadin protein, reducing immune responses, but the gliadin is the protein that enables the gluten that is essential for properties required for bread making. how is this reduction of the gliadin protein impact the bread making properties in the modified wheat? So I guess, can you still make bread if it's gone?
Oh, yeah. I've never tasted that wheat, because even that would be illegal in Europe. Like, but because you cannot even grow this wheat, and can even make bread in sufficient quantities. But it actually has been done. So it's a group in Spain which has been doing that. And according to what I've seen in presentations, I mean, there's still sufficient gliadin cell to make what looks like a normal bread. How it tastes, I don't know. It's but it's indeed it's correct. Like, that's other examples. Like, is, for example, cassava, which is really a primary food product for many people. And like in Africa, I mean, they produce actually very toxic molecules which have to be removed. Like, and it causes quite some problems. Well, with gene editing, you can knock these genes out, like, and having cassava, which is much better suited for human consumption. And there will be many of these examples. Like, and that's maybe as a last sentence, I would really add to the discussion. I mean, I think GMO, like, I mean, we have a legislation. I think it will be very difficult to change this in the near future. I mean, it's so much in the brain, like, of people, like, has been associated with certain things. Genome editing is different. It's actually different from the GMOs. It's a breeding tool. You make the same mutations, the same changes as you do with classical breeding, only faster and much more precise. And we have to make a distinction between these things. And I think we have a new chance, like, in fact, with this technology to bring benefits to our society. And let's not paralyze the discussion by putting it in the GMO box, because that's not where it belongs.
Okay, so let me pose a last question, because I don't want to appear as being suppressing this, because it's critical of public media. So let's pose that as a last one. According to my perception, the question is: the population is informed one-sidedly, especially by the public media. Has the influence of media reporting on this topic already been studied? I guess this goes to Angela.
Yes, there is a lot of research that focuses on the way media represents this issue. And of course, what we read in the newspaper is going to impact how we see it. It's also going to create associations. I'm just going to give a simple example. Frequently, not so frequently now, but in the past, whenever newspapers were reporting about gene technology, they were using some stock photo with an apple and a syringe, or some sciencey guy in a lab. And that's creating associations in people's minds that they use then when making decisions or responding to the questions we ask them. So I think this has increased sort of this idea of being careful about the way media presents things, and kind of associations are created. But then also we have less and less science journalists here in Switzerland, or I suppose this is something that goes globally, that because we don't have any journalists that know about science or have a sort of background in science. And this, of course, makes the journalistic style then a bit different. And it, of course, sells better when we say there is a risk than if we say we can pretty surely say there is no risk. And there's definitely a difference there.
Maybe one add-on question to Matin with asking for a short answer, because we're really closing in. You know, you worked a lot in the global south. Is this discussion in the developed countries, which is very critical of all these developments, is that kind of influencing strongly opinions of the people in the global south as well?
Yes, it is. I mean, of course, only by those who are relatively well educated, but there things spread in the newspapers and in the media similar to what we see here. I know that from India, where I've worked a lot. I know that from Kenya, Tanzania, Uganda, where I've worked a lot. So this is influencing, and you see the same types of pictures in the media that are trying, you know, to portray a picture of really unpredictable risks. And of course, this is influencing the people who read these newspapers and the discussion and the fears. And therefore, we cannot say that it's only in Europe where we have these kinds of perceptions, but there's very solid research that shows that things are influenced to a large extent by what's happening in Europe, because it may be the same journalists that are either having their connections to Europe or that have studied in Europe or look at Europe, and with the social media and the internet, of course, connections are very easy to tie and identify.
Okay, thank you so much for this really lively Q&A session. Thanks to the audience for all their interest, for all their great questions. And I'm sorry, I think we tackled many of them. There are some of them not answered yet. Maybe you can try to contact the speakers maybe by email if they're willing to do that. Was just not enough time to cover all of them. The next webinar of this series, CRISPR and somatic gene therapy, will be on Wednesday, April 28th, and the fourth and last one, CRISPR and society, will take place on Tuesday, May 18. Please visit the Q&A box to find the links to these two webinars and a short poll asking for feedback concerning today's webinar. So if you have to, give positive feedbacks, but also criticize for whatever you didn't like, please do so. Thanks again, the speakers, for their insights and engagement with the audience, and to all of you for your interest and the excellent questions. Thank you and goodbye.
Yeah, thank you, Thomas. Yeah, bye.